Hello students, welcome again to the second lecture of the growth and development series. Last time, in the introductory lecture we also saw the process of spermatogenesis. So now we continue with oogenesis. Now when we consider this process, remember we have to consider both these components. That means the process of formation of a mature ovum from a primordial germ cell and the process of development of the ovarian follicles, which happens in the ovary. So in the picture, this is a cut section of the ovary where all this is going to happen. Okay, and then we are going to look at the two processes that we will have finished by comparing and contrasting them, and then some factors that have affected can affect gamete transport. Gamete, means sperm or ovum transport. And then what happens in if there are gametes with abnormal chromosome numbers, so how that can happen, and examples of some clinical syndromes that can occur because of abnormal gametogenesis. Okay, so here is a cut section of the ovary. Let's learn by and large as a schema in a schematic view what's going to happen in this process of oogenesis. So firstly, if you look at the structure of the ovary, it is surrounded by a fibrous capsule known as the tunica albuginea. Now underneath that there is like a simple cuboidal epithelium known as germinal epithelium, which actually is a misnomer, means it is misnamed and has no functional significance. It does not contribute to any of the germ cells in the ovary. Okay. Now in the outer zone of the ovary you can see a lot of things happening. So there are some follicles maturing, and then you get a mature grafted follicle, and then this bursts or ruptures when ovulation happens, and then something else is left behind. So all this is happening in the outer zone of the ovary, which is known as the cortex. The inner zone of the ovary you can see is very vascular, so all the blood vessels, lymphatics, nerves, etc. will enter that, and this central zone is known as the medulla. Okay, so let us see more about how this story develops. So the story of oogenesis begins before birth in a female embryo at just 2 weeks intrauterine life. So the primordial germ cells, as in spermatogenesis you saw, have migrated to the wall of the yolk sac. From there, they will migrate into the embryo and take up residence in the developing gonad. In fact, they induce the development of the gonad, like as if they go there and build their own house. Once they have lodged in the developing ovary, they undergo mitotic divisions to form many oogonia. These undergo further mitotic divisions and then differentiate, that means they grow larger and mature, into primary oocytes. So even at fifth month of intrauterine life, in the fetal ovary, there are about 7 million primary oocytes. So they will be all lodged here in the cortex. Now, these primary oocytes, so that's like any typical cell, get surrounded by a single layer of flattened cells, as you can see in this picture. Now, where do these cells come from? So they're derived from the ovarian stroma, that means the connective tissue of the ovary. Okay, and these are the other cells, the other stromal cells that are surrounding it, undifferentiated. So such a structure is known as a primordial follicle. What does it contain? It contains a primary oocyte, and remember, it's before birth. Now, at birth, what happens? Primary oocytes begin the next stage of division, and so they begin the prophase of meiosis 1, but they don't complete it. Okay, by birth there are about an average of 600 to or 6 to 8 million. Okay, now prophase of meiosis 1 means where the chromosome material, the nucleus, has just started to kind of get together to form thick chromosomes. Now, this prophase of meiosis 1 is not complete, but it gets arrested in the diplotene stage of prophase 1 till puberty. That means till a number of years. You know, puberty age is about 10, 11 onwards. So till then they are arrested in prophase 1. And if you want to be specific, diplotin stage means the double-stranded chromosomes are seen. Now, how is this controlled? This is regulated by a factor known as OMI, oocyte maturation inhibitor. It's a peptide or a protein produced by the surrounding follicular cells, which inhibit the further division of the uh, primary oocyte. Now, during childhood, what else happens? These primary oocytes, even if they are arrested in prophase 1, many of them will degenerate until by puberty only about 40,000 primary oocytes will remain. And during a woman's reproductive lifespan, only about maybe 500 will have a chance to ovulate. Or so you can imagine how drastically the number is going to be reduced. So here is another picture of a primordial follicle. So remember this definition. It contains a primary oocyte and it's surrounded by a single layer of flattened cells. Okay, and this is a actual picture. You can see the flattened epithelium and the nucleus of the primary oocyte in the ovary. So let's see now further development of the follicles, or folliculogenesis as the process is called. Remember, the primordial follicles with their primary oocytes exist in the same condition until puberty, when under the influence of hormones from the pituitary, for example, follicular stimulating hormone, now these follicles undergo further development. So every month, about 15 to 20 follicles begin to mature and pass through the following stages. A is primary or pre-antral follicle, as it's called, or secondary or antral follicle, and Graafian or mature or tertiary follicle. Now look at the pictures. So at puberty, look at what's happening to these flattened follicular cells. They become simple cuboidal. So they're increasing in height or size. And they begin to secrete this pink membrane, which in the next stage you can see is become quite thick, and hence now we call it the zona pellucida. This protects the growing the oocyte inside. Now look at the follicular cells. So when the follicular cells become cuboidal, we call it an early primary follicle or a growing follicle. And then these cells become now multi-layered by dividing. Then it is called a primary follicle. Remember, it still contains a primary oocyte. Now the second stage is the secondary or antral follicle, which is shown in this picture here. Antral means cavity. Now see, it becomes a multicellular layer of follicular cells. Soon small cavities start sorry start appearing between these follicular cells. First, they are small multiple cavities. Then they soon start fusing and becoming one single cavity, as we shall see in the next slide. So here, this is a secondary follicle. So again, oocyte, note the pink zona pellucida, and these were all the multi-layered follicular cells. Surrounding this is all the ovarian stromal cells. And in these follicular cells, you can see cavities developing, and then they are fusing to form one cavity. And the follicular cells also start secreting some fluid inside, that is known as follicular fluid. Okay, hence this is called the secondary or the antral follicle stage. Now this follicle continues to enlarge. How is by increasing amounts of follicular fluid is secreted by these cells. Hence the cavity begins to enlarge, and all those follicular cells then undergo a rearrangement as seen in this picture. So see how large the antrum or the follicle is filled with follicular fluid. Now one group of cells gets pushed to the periphery, and these are going to be known as membrana granulosa cells or granulosa cells. Now look at the oocyte surrounded by the zona pellucida is pushed to one side of the wall of the cavity, because this is enlarging with lots of fluid inside. So let us see what these orient ourselves to some of the names of cells surrounding this oocyte. So you have the oocyte, then the zona pellucida, And then look at these layer of cells immediately surrounding it. They are known as corona radiata. Corona means sun, radiata means radiating rays of the sun. And then there is a heap of cells which attach this group of cells, oocyte and this group of cells, to the wall of the follicle. This group of cells then, like a stalk, this group of cells is known as cumulus oophorus. Again, cumulus means collection and oophorus in the follicular cells. Now, so far the stroma was quiet. Stroma, I mean all these cells around it. But here note, now outside the granulosa cells, the ovarian stromal cells, that means those connective tissue cells, also undergo differentiation to form two kind of coverings. The inner covering is known as theca interna. It is vascular, okay? It's quite vascular and cellular layer. And the outside capsule, like a covering, final covering, is known as theca externa, which is more fibrous, it means more connective tissue cells like that, okay? So theca interna is more vascular out of the two and more kind of cellular, whereas the outer one is more fibrous. So here in this picture now, unlabeled picture, can you make out two types of follicles and guess what they are? So what is this? This one matches this upper one, right? So identify this follicle and the structure seen there. Now this is a Graafian follicle. So again you see oocyte, zona, looks like a featureless membrane, corona radiata, cumulus oophorus, by which the oocyte remains attached to the follicle wall, and these are the membrana granulosa cells, and then the theca interna and externa will be outside. So remember, many follicles start to grow, but usually only one follicle in each ovary reaches maturity. The others, what at whatever stage they are, they might be like primordial follicles, primary follicles, or secondary follicles, will start degenerating. Now remember, there are two ovaries, but all this, each cycle usually um, occurs in both ovaries, but ovulation, that means release of this oocyte, we will learn in the next class, will occur from one side ovary only. More will be explained with when we do ovulation. Okay. So here is again a structure, nice structure of a Graafian follicle for you to revise. Note now all the parts, and here you can see the vascular, theca interna, and then will be the externa. Okay. Now, what this contains, now here is labeled as primary, but when we do further development of the oocyte, you will understand actually what it contains just before ovulation. Okay, again, all this is a close-up picture of the oocyte with its zona and corona and the cumulus oophorus. Okay, so I'm sure now you can identify all this. So, summary of what was this folliculogenesis phases. Summary of development: we started with a primordial follicle which became a primary follicle. When cavities developed, it became a secondary follicle, then it became a mature Graafian follicle. Now we will see what it contains by the time when it becomes mature. And then ovulation means this oocyte is going to rupture and come out of, as you can see in this lower picture, is going to come out of the Graafian follicle, and what's left behind is something called the corpus luteum. Of it'll be like a cellular mass. The cells show yellow pigment, so it's called luteum, means yellowish, and then corpus luteum, and then a scar-like structure called corpus albicans. So all this you will understand soon. So at puberty now, there were about, remember, 40,000 primary oocytes. So let us now look at, while all those follicles are doing their own story, let us see what this primary oocyte is doing inside that follicular wall. So phase 1 of oogenesis has already begun before birth, where primary oocytes have formed, and they were arrested, remember, in prophase 1. If you want to be specific, diplotin stage of prophase 1. Now, at puberty, the phase 2 begins. Okay. So what happens, wherever, at whatever stage this primary oocyte was arrested, now it completes the whole meiosis 1, just like, remember, in spermatogenesis. So from primary oocyte, we get a secondary oocyte. And so here, you can see Okay, I'll explain from the figure later on. Just understand this. So from a primary, you get a secondary oocyte and a first polar body. Okay, all this, remember, is happening while this is inside the still inside the Graafian follicle. Now this kind of division, meiosis 1 is a reduction division. So what happens to the chromosomes? The chromosomes were 2n or diploid, right? Now they become n or haploid number of chromosomes. Now let's see here. So in the figure, this line depicts that whatever has happened here is before birth. Remember, primordial germ cells, oogonia, many oogonia, and primary oocyte. Okay, before birth. Now at till this pink line here, see, all this is before birth. So primary oocyte with 2n number of chromosomes. And remember, before anything undergoes cell division or meiosis, it has doubled its DNA. So amount of DNA is 4n, but chromosome number is 2n. Chromosomes become just thick and double stranded because of the double amount, the tetraploid amount of DNA. We explained that in the first lecture. Now, after birth, what happens? This, under the influence of hormones, at puberty, meiosis 1 continues. So, this primary oocyte now becomes undergoes reduction division to form one secondary oocyte and one polar body. So you see, this is an unequal division. But what is unequal? Is it the nuclear division or it is the cytoplasmic division that is unequal? What do you think? Because here we see one N and one N. So it's the cytoplasmic division. Why? We will learn later. Okay. Now you get a large one secondary oocyte and first polar body. But this secondary oocyte begins meiosis 2 just like in spermatogenesis, but it gets arrested in metaphase 2. That means where the chromosomes come near the equator, and it just gets arrested there. Okay, and as such, so it's not still a mature ovum. It will be released as such at ovulation from the mature Graafian follicle. Now, okay, so this will be released. Now, when does meiosis 2 get completed and when does it occur? So that's the phase 3 of oogenesis. Only if fertilized, remember this, only if fertilized, this secondary oocyte which was arrested in metaphase 2 will complete the second meiotic division to form a mature ovum. That's here. See, so fertilization only, it will form a mature ovum, again a large cell, and a smaller second polar body. So now what will happen to the DNA and the chromosomes? So here in meiosis 1, remember this was halved, right? 23 and 23 went into the first polar body. But the amount of DNA, because here it was 4 times n, here it is still 2n, right? So another division must occur. Now at the end of this, when the mature ovum is formed, it is haploid amount of DNA, haploid number of chromosomes, or 23. Let's see further. So this explains it simply. All oogonia, all this before birth, primary oocyte, then at puberty undergoes reduction division to form secondary oocyte with n number of chromosomes and first polar body. But remember, this is double amount of DNA. Now that will undergo meiosis 2, which is like mitosis. So you get uh, uh, ovum and second polar body. But remember, this is formed only at fertilization. So one cycle of oogenesis begins before birth and is complete only if fertilized, only at fertilization. So majority ones, you in a lifetime a woman doesn't have 400, 500 children, so majority will degenerate at whatever stage they have to be, uh, you know they were developing. And these pictures just show ovulation, how that oocyte surrounded by its different cells will release. We will see more about ovulation in the next lecture. Okay. So I hope you understand the concept of meiosis 1 and meiosis 2 in oogenesis. If you still have doubts, you can ask. So now here we are. All this was happening in the ovary. When ovulation occurs, the what is released? It's the secondary oocyte arrested in metaphase 2 is released and it's caught by the fimbria of the tube. Suppose this side ovary is releasing. Okay. Now How will this ovum, the secondary oocyte be transported to the site of fertilization? Where is the site of fertilization? In the ampulla. That's the broadest part of the tube. So how is that moved there? So there is uterine epithelium. You know what kind it is, if you remember? It's simple ciliated columnar epithelium. So the cilia help the oocyte to move towards the site of fertilization, plus uterine tube wall, smooth muscle contractions, secretions, etc. Now, how do the sperms come into the female genital tract? Obviously, they're deposited in the vagina, move up into the cervix body, but they are very, very motile. So they have their own motility plus uterine contractions, and they will migrate to the site of fertilization. Now, if fertilized, then remember that secondary oocyte becomes a mature ovum, and after sperm has entered, that cell will be known as the zygote. We will see that all those separately, and now this structure is moving back into the uterine cavity to stick to the wall of the uterus, that is the endometrium, and then it will get implanted to grow further. Okay, so what factors could affect this transport in the tract? Obviously, first the tubes must remain open. If there is a narrow fibrous tube, nothing will move. Then the cilia epithelium must be intact, and secretions must be adequate, and the sperm should be normal, motility, contractions should be normal, etc. Okay, so summary of oogenesis: it begins before birth, and then from puberty to menopause. Menopause usually occurs, it depends on race and countries, etc., but usually between 45, 50, some stop at 55 also. So from puberty to menopause, every month usually one primary oocyte arrested in prophase 1 completes meiosis 1 to form a secondary oocyte and a first polar body. It may happen from either ovary. They start developing in both ovaries, but only in one ovary, or it will go to this stage and a mature graphene follicle will rupture. um, you might think of what happens in twin pregnancy, so those we will deal with separately later on. So at ovulation, what is released is important to remember. It's not a mature ovum, it's a secondary oocyte arrested in metaphase 2. Only if fertilized, the secondary oocyte completes meiosis 2 to become a mature ovum and a second polar body. So one cycle of oogenesis hence begins before birth and is complete only at fertilization. The two polar bodies usually degenerate. Sometimes, one continues to divide, you might get three also, but they degenerate. And one primary oocyte finally then gives rise to one ovum with haploid number of chromosomes and haploid amount of DNA, and majority will degenerate at any of the earlier stages. So knowing this, now you can compare both oogenesis and spermatogenesis. When does spermatogenesis begin? We say, though primordial germ cells are there in the embryonic testes, but they don't do anything before birth. They only begin to further differentiate at puberty. Whereas in oogenesis, it begins before birth, and at birth, ovaries contain primary oocytes, whereas at birth, testes contain primordial germ cells. Uh, Oogenesis is limited because it will begin further at puberty and only during a reproductive lifespan of a female, whereas spermatogenesis continues throughout life. So even a 90-year-old continues to produce sperms. The one cycle is much shorter. It takes about 64 to 74 days, and whereas one cycle takes many years. Now, from one primary oocyte gives rise to one ovum, whereas one primary spermatocyte gives rise to four haploid spermatozoa or sperms. So what is a normal sperm count? Is usually 300 to 600 million per 3 ml of ejaculate fluid. Per ml, obviously it would be 100 to 300 divided by 3, right? So you can remember it like that, per 3 ml. Now, majority, uh, these primary oocytes remain, as we've said. So this is all repetition, okay? Are arrested as primary oocytes. Only fertilized, then they go further. Now, all ova have, what is the genetic combination? So all ova have 22 plus X chromosomes, whereas half the sperms will have 22 plus X plus, and half will have 22 plus Y. So who determines the sex of the embryo? Again, recall. Now, here you note this is spermatogenesis, and note that the division, both cytoplasmic and nuclear divisions, are equal. So primary to secondary spermatocyte, secondary to spermatids, then they undergo change to form four spermatozoa, right? But in oogenesis, see, this is oogonia I've already finished. So primary oocyte gives rise to, undergoes meiosis 1, secondary oocyte and first polar body. See the unequal cytoplasmic division. So the nuclear division is equal because n number of chromosomes, 23, go to the polar body also. So why do you think this is unequal? Now secondary oocyte, if is released, and if it fertilizes, this gets becomes a mature ovum. Again, see the unequal division. It's a large cell, and the polar bodies are small, and they're going to degenerate. So the polar bodies, if you notice, are there just to get rid of the excess nuclear material. Okay, but why is all the cytoplasm retained here? So see how complementary it is. Remember the sperm, it lost all its excess cytoplasm as Raynaud's body, because it needs to be very fast and thin and motile, so it can't carry excess baggage. So the ovum needs to be huge and fat, carrying all the organelles and nourishing material initially to be able to sustain that oocyte, or sustain the zygote after fertilization. So actually, when a sperm approaches an ovum to fertilize it, it's like a rocket, a pure, approaching a huge planet. It's something about 85,000 times larger than that sperm. So that's why it retains the cytoplasm. Now, we've seen normal gametes, but as always, something can go wrong at any stage. So sometimes, if the primary oocyte and inside early divisions are not normal, there are some gene mutations, you might get a primordial follicle containing two oocytes, trinucleated oocytes. These will usually degenerate. Similarly, sperm abnormalities are more many. So you can have two-headed sperms, two tail, short tail, very long tail, small head, large head, abnormal head, bent head. And here you can see in this kind of a slide, so you can examine sperms under the microscope. You can obviously see some are not normal. See, so they have to have kind of a normal appearance. By experience you can make out some only head, some very short bifid tail and all that. So this is a normal sperm, and this is a grid. Like under the microscope it will appear like, if you put a slide underneath, it will divide the slide field into squares, and in each square then you can count the number of sperms, and also not only number but see their structure also, because these abnormal sperms are incapable of fertilizing. Okay, now let's see how gametes with abnormal number of chromosomes can result. So this is known as non-disjunction, or failure of chromosomes to separate either in meiosis 1 or meiosis 2 of either spermatogenesis or oogenesis, doesn't matter when. So this can result in gametes with an abnormal number of chromosomes, either one more or one less. So let's understand how this happens. Now here, this is only showing you a normal meiosis 2 division. Means we presume that meiosis 1 was normal, and the reduction division happened with half the number of chromosomes going to each cell. This was normal. Okay, now second meiotic division also was normal, so there's a split in the centromere, and each one gets a single one chromosome, which becomes haploid. So haploid and haploid, right? And the DNA amount is halved. This is normal. So there are 23 chromosomes. Now, if non-disjunction occurs in the first meiotic division, that means each of these chromosomes, just like here, was supposed to go separate ways in each cell, say primary to secondary spermatocyte or oocyte, but they both went to one cell. So there is one cell which has one less uh, number of chromosomes, right? Now in the second meiotic division actually occurred normally, but now here this one has extra, so they split and they went to the two daughter cells. So in effect the two daughter cells got, final two gametes got an extra chromosome, n plus 1, whereas these continue to divide with one less chromosome, so they were 22. So this can be, I'm giving you an example of say chromosome 15 pair happens like that, right? So the total number then would be extra or less. Now this non-disjunction can occur at the second meiotic division. That means at this place. So now normally they're supposed to split like this. Now if they don't split and it goes to, both go to one cell, the other one doesn't get, and so on. So again the end result is extra or less number of chromosomes. Now if these abnormal gametes fertilize, suppose a sperm with a 24 chromosomes fertilizes an oocyte with normal number, then what will be the result? Will be one extra chromosome in the zygote. Okay, and that will can result as a genetic syndrome, or even a gamete with a less number will have, finally the zygote would have less number of chromosomes. So what is a chromosome spread? When you can examine these chromosomes under a slide, you can cell culture and spread them and all that. That's known as a normal karyotype. So each cell has 22 pairs of autosomes and one pair of sex chromosomes. Now they are arranged. This is called a fish technique, fluorescent in situ hybridization technique, where you can actually color these chromosomes, and you can also see the different regions in different colors. So mutations or abnormalities can be easily detected. Don't worry about the process, or you may already know it. But now these chromosomes are arranged in decreasing order of size. So they are pairs and decreasing order of size until the last are arranged. Those are the sex chromosomes. So here you can see Y chromosome is very small, X is a big chromosome. So if a karyotype reads as total number of chromosomes are 46, and we write it as 46 comma XY, it doesn't mean 48 chromosomes, it's just written 46 totally including the XY, but what type are those? So XY. This is a male karyotype. Now look at this here. Again, they are arranged in decreasing in pairs. These are the autosomes, 22 pairs, and the last one is the pair of X chromosomes. So that is 46 XX. So that is a female karyotype. Here there is one, but the other pair is gone here in the spread, but it's okay. It's normal only. Okay. So now what about an abnormal number of, if the non-disjunction has happened either at meiosis 1 or 2? For example, you see, now it may not affect, it may affect only a single chromosome, or it may affect more than one chromosome pair. But here we can see examples. So all this seems normal. So you have to examine each chromosome pair and come down, down, down. Now look at chromosome 21. There have to, 3 have come, right? So plus 1 where it shouldn't have. That means another gamete could have got one less, right? And what is it now? It's a male karyotype, because there is a small, sorry, this is no Y, there is two X's, right? So it's a female karyotype. And how will we write this abnormal karyotype? So we write it as 47 because one extra, XX because female, and which one is extra? We write as plus 21. So suppose 15 was extra, we would write plus 15, or one was less, we would write minus 16, and this would be like 45XX. So whatever is the abnormality. Now because it's 3 chromosomes, this is also known as trisomy 21, and very well is also called Down syndrome. So again, because which fish techniques, they also, you can in the nucleus, they can tag these abnormal chromosomes and with a particular color. So normally you can see the red should be 2, like 2 pairs, right? But here the red is 3, which is chromosome 21, and it can manifest as Down syndrome with a lot of abnormalities. We are not going into that. Okay. So if, how does this happen? So gametes, one gamete must have had an extra one. Okay, and this unites with a normal gamete. The result is an embryo with an extra number of chromosomes. Okay. Another example, trisomy 13. So now notice, now this is all pairs, but number 13 has 3. And what kind of, again, is it a male or a female? So here you can see again 2X but no Y. So again it's a female embryo. This is of course incompatible with the survival, because a lot of abnormalities depending on the genetic condition. So this is also written as 47XX plus 13. All right. Then sometimes chromosomal anomalies may involve the X or Y chromosome, means numerical number. Now, one example is Klinefelter, where there's an extra X come in. So that's XXY karyotype. So we would write it as 47, comma XXY. Or there is Turner syndrome. You must be learning all this in genetics. And Turner syndrome is a deletion syndrome. So there is one less X. So we write it as 45XO. Obviously, both will manifest differently. So this is usually in a female because I mean sorry a male because XY is there, but an extra X is there. So it's like a feminine tending towards the feminine characteristics, gynecomastia, um, poorly developed sexual characteristics, will be sterile, infertility, small testes, etc. Whereas in a Turner syndrome, usually affects a female because one X is missing, and she'll be of short stature. Again, poorly developed secondary sexual characters, streaks, fibrous ovaries, so she'll be sterile, and other things shown here. Now, I'm not discussing structural chromosomal anomalies. Remember, structural is that the chromosome number will remain normal, but a part of a chromosome may be, missing, or one chromosome may go and attach to the other. So those are those translocation syndromes. These are just examples of how number of chromosomes may become abnormal during non-disjunction. So I hope something is clear here, but if not, you can of course post your queries, and definitely we will have some kind of interactive time either during your lecture hour. We can have some synchronous lectures, but I just want the first 2 3 weeks to settle down because we are having very chaotic time with other timetables, with students coming back to campus. So I will let you all know which lectures will be live synchronous. Okay, so just patience for now. Try and go through them, and you can post your queries by email. Okay, thank you.